Same time, 12 noon, Orit Ben-Zvi is going to be speaking about systems thinking, and Tali's going to be hosting her. The STEM education activity here at MSU, we thought that his work would, definitely spur some lively discussion for us. And so today he's going to be talking about measuring conceptual change in molecular biology. So welcome, Brian.
Thanks, Becky. And thank you, everyone, for inviting me. It's a real pleasure to be here. I guess I suspect there's something going on. The red is not showing up in my presentation. And you might know that Nebraska has a certain affinity for red, so I guess Michigan State, you got this one, so we'll get you next time. So I actually have a lot of friends from Michigan State. I couldn't help but show this. So as you are aware, the alumni here are quite passionate about their institution. And I recently found when one of those friends babysit your daughter. So this is my daughter, this is my friend, and as you can see, he's forced her into this Michigan State outfit. But what you'll also note is that despite this, she has still gone back to her Nebraska football. She is still clinging to it as a loyal Nebraska fan. All right, so today I'm going to tell you about my efforts to develop a programmatic assessment for molecular biology. Just to give you a little bit of my background, so I did a traditional molecular biology, biochemistry PhD at Yale. I then did a biology education research postdoc at CU Boulder. And I'm now at Nebraska as an assistant professor where I run my own education research group. The work that I'm going to tell you about today pretty much occurred at University of Colorado Boulder. So when I talk about our program, I'm talking about the University of Colorado's molecular biology undergraduate program. So as you are all, I'm sure, aware, there's been many, many calls for educational transformation over the past few decades. These reports come out every year or two. They typically discuss a few points that have sort of been well hashed at this point. So one is that they all emphasize the importance of undergraduate science education. What we do matters, both for economic reasons, also for cultural reasons. I think of college faculty as sort of being the interface between the general public and the research world, and so the way we teach classes really influences how science is viewed and practiced more largely. Several of these reports outline what students need to learn, what's the type of curriculum that we should be building for students to succeed in sort of the modern workforce. They all, in one way or another, promote the adoption of what I'm going to very collectively and generically call interactive teaching methods. And, of course, we're aware that there has historically been underrepresentation of particular groups, and that that's something that we need to investigate and remedy in the future. So this is all to say, one thing that's not really touched on as much in these reports is assessment. But assessment really forms the backbone of all of these efforts. In order to do any sort of evidence-based teaching or any sort of research on teaching, really you need some type of assessment, some type of readout of what is actually in students' minds. Just also to make you aware, in the field of biology, we have a particular report that has risen to prominence. So Vision and Change is our most recent consensus report. This is a report that was the result of conversations between more than 500 different biology stakeholders, all the way from students to faculty to administrators, policymakers. And this report did two important things. So one, it outlined 5 core concepts that biology programs can be structured around or should be structured around. And it also identified 5 core competencies or science practices that biology programs should also be oriented around. Of course, lots of departments across the country have had responses to these calls. They've all done it in their own unique, different ways. At CU, we had the Science Education Initiative, which was essentially a university-based effort to provide departments with the resources they wanted to do the educational things that they wanted. So it was very department driven. What this program did was it gave the department funds to hire people like me or like Anne Marie or like Danny Caballero to come in and do course-based transformations. And so up until that time, my department, molecular biology, had spent a lot of time developing learning goals for their core courses in their introductory series and all the way actually up through their advanced capstone courses. They had implemented a variety of different learning activities and developed multiple assessment tools for use primarily at the introductory levels. When I came in, the department wanted to think a little bit more broadly about how these various efforts were affecting students as a whole and across the entire curriculum. So the question that we wanted to address was, what do biology students actually know when they graduate? So here's a very generic pipeline of college. Students come in like this, sort of naive. They take 4 or 5 years of classes. They exit here with a fancy hat and some sort of credential that we take to mean that they've learned something about biology. What that exactly is not always well defined. And I would also argue that most departments currently don't have any mechanisms in place to actually assess what biology students know when they graduate. Really, the only thing they're sort of asked is, have you checked all these boxes, and have you paid all your library fines, etc. You're good to go. And the reason this is problematic is that we don't really know what our program is producing. So do students actually remember things that they learned here? Have they put these things together in a way, or can they apply these things to more complex systems? And then also an important question is, can they demonstrate knowledge without the benefit of having studied the night before for a test that's sort of telegraphed, that the instructor is giving you very clear language on what you should be expected to be tested on. So there are a lot of things that I would like to know about graduating seniors. We'd like to know how their fluency with the process of science, their ability to form arguments, the affective dimension, their attitudes towards science, their identities, all these different things. For the purposes of this talk and for my project, we focused specifically on conceptual knowledge. And the way that we went about assessing conceptual knowledge is through developing an assessment that we now call the Molecular Biology Capstone Assessment. So the Molecular Biology Capstone Assessment was designed explicitly to identify areas of conceptual difficulty. So our process is going to be oriented specifically to finding these areas of conceptual difficulty and estimating how prevalent they are in the population. We had to delimit our scope, and so we're going to be focused primarily on molecular, cell biology, and genetics. So whereas most concept assessments or concept inventories are relatively narrowly focused on an individual topic or maybe a course, our scope was very large. So it's entire fields, which obviously there's limitations to what we can actually assess across such a large domain. And finally, most standardized concept assessments have been developed for introductory level courses and introductory level students. This is really among the first that targeted upper division students explicitly. And also, to my knowledge in biology at least, it's the first that targets an entire program rather than a discrete course or topic. So of course, there were issues and struggles that we knew we were going to be facing, but we also saw that there were going to be many benefits. The first was that the process of developing the assessment, determining what things should be on the assessment, what are the concepts that students should know, having those conversations among faculty we thought would be beneficial for the department. We also hoped that it would provide some amount of information about students and what they've learned as a product of our education. We also set out from the beginning that we wanted to develop something that could serve as a tool for other similar departments. And so we were somewhat constrained in terms of what we could build, and we wanted to make sure that whatever we built could be used elsewhere. And finally, I think this was maybe not as much of a goal at the beginning, but it was something that we really came to realize as the process went on, which is that programmatic assessment is just so uncommon. It's really not something that's done. And so our sort of first generation efforts in this direction to think about assessing students at a programmatic level, hopefully our efforts can help pave the way for more thinking in this way. Of course, there's a number of challenges. Most of these challenges stem from the way that undergraduate majors are structured. So students come in here, they take a series of core courses where one is typically the prerequisite for the next. And so you have this nice pipeline and you have a content that is fairly narrow. The problem comes when you students go into the upper division classes and they start taking electives and they sort of go all over the place. They start developing subdisciplinary specializations, they start missing particular courses and taking other courses. And this creates a lot of problems. So the first is that students differentiate. And so in thinking about the content on the assessment, you have to account for this. You have to strike a balance between what are the things that we think our students should have gotten regardless of what classes they've taken versus what are the things that students, we'd like them to get, but they might only have gotten it if they took a particular upper division course, and we're okay with that. So we attempted to solve this problem by focusing on things that we thought were core concepts that most or all introductory biology courses would cover in some way, and that would be reinforced through their application in upper division courses to more complex biological systems. Another very pragmatic issue, there's really, at our institution at least, there was no departmental mechanism for administering exit assessment. How do you actually get these students into a room, into some space where they take the assessment seriously, and you can collect them all. You can't just send emails out to random seniors. They're not going to take your assessment. We at University of Colorado, we have Capstone courses. Students are all required to take one of 3 different Capstone courses. And other institutions have similar courses like this where they have a high proportion of seniors in some upper division course. So we targeted our assessment to be given in those courses. This at least gives us a way of getting the test to students and a way of giving them some credit for actually taking the test. But since the scope of the test is beyond any one of those individual courses, we really can't hold students accountable in the same way that we would for a normal course test. So if they haven't covered that material in class, there's sort of this, implicit contract with students that we're not going to test you on things and give you points for things that you haven't explicitly covered in this class. The way we solve this is that we gave students participation credit for attempting the assessment, and that's really, so it's everything we do is going to be under low stakes conditions. So in order to develop this assessment, we followed the concept inventory model, which has been fairly well characterized by Wendy Adams and Carl Wyman, and that has been used quite a bit in biology and a few other disciplines to develop assessments. So I'm going to walk through each of these steps, but we start by identifying the learning objectives that we actually want to test. We then go through a process of collecting student thinking to figure out what students actually know or what kinds of different conceptions they have with relation to these learning objectives. We develop questions. We go through a very iterative process of showing these questions to students and faculty and revising the questions in order to make them more clear and more scientifically accurate. And then finally, once we have questions that are stable, we can pilot them and give them to a lot of students and see what types of measurement characteristics they have. All right, so the first step that we embarked on was to identify important concepts and learning objectives. In order to do this, we took a multi-pronged approach. So we did look at the current course learning goals for our own curriculum. This was something that had a lot of effort put into it, and we were able to cull some ideas from that source. We did a little bit of looking at relevant textbooks, but that was by no means a major source of ideas. It was more of a confirmatory source. The main bulk of our learning objectives came from faculty interviews. So we sat down with, I think it was 22 individual faculty, and asked them questions like, what are the things that you think every molecular biology student should know upon graduating from our major? What are the things that you'd like molecular biology students to know and potentially be able to do if they were to join your lab after graduating from our major? What are things that you think students particularly struggle with in our major? different angles to try to get at what they value, what kinds of concepts and objectives they value in our students and our curriculum. We also had a series of faculty roundtable discussions where we sort of vetted these ideas, tried to gain some clarity on what they actually mean, and distill them down to a manageable list. So from this whole process, we came up with 18 different concepts and learning objectives, and I'll give you an example of what one of those looks like. So at the concept level, we might say that individual molecules move through liquids as a result of thermal motion and random diffusion. And a linked learning objective, an actual testable statement, would be we would want students to be able to explain how a molecule can travel between cells that are located multiple cell lengths apart. Now, I'm also conscious right now, Melanie, you might be cringing. They are not actually going to be explaining how a molecule can travel between two cells. But we're going to use an alternative question format as a surrogate for an explanation. So when we started this process, and I should also add, I forgot to add, so Caleb Trujillo, who's now here as a postdoc working with Tammy, actually started this project while he was an undergrad at CU. And so I sort of came on after he left. So we came up with this list of 18 concepts and learning objectives. And at the time, Vision and Change had not come out. So Vision and Change came out shortly after we developed this list. And so ideally, you would have started with Vision and Change and flowed from that, but we sort of had to retroactively align our concepts and learning objectives to Vision and Change. And what we found, we used a sort of a faculty-driven process. We found that we have pretty good representation in evolution, pretty good representation in pathways and transformations of energy and matter. The bulk of our questions are in these two, under these two core concepts. And we have relatively few questions that really capture systems. And I think partly that is because systems is a particularly ill-defined area within biology right now. And a lot of what one person might call systems, we ended up classifying, and others ended up classifying as information flow, exchange, and storage. So that's something that I think the biology community needs to continue to sort out. So we have our core concepts and learning objectives. We then administer these questions, or these we administer open-ended questions to students in think aloud interviews, where they're given a question, they generate for us a number of different ideas. So following up on this last example, the question might be, well, how does a molecule travel across a fluid-filled space to bind to a receptor protein? And students will give us a number of different examples. So they might talk about diffusion, random processes, and molecular collisions as how something ends up encountering something else. Some students will talk about transport by motor proteins, which is something that does occur in biology, but only in particular places. They'll cite electrostatic attractions, but not necessarily appreciating different fields that are present in biological systems, the effects of water, and what distance these electrostatic attractions are actually meaningful at. And then finally, there's some sort of naive conceptions where people think that molecules have some sort of intuition about where they need to go, and the receptor sort of pulls the molecule in because it wants to bind to it. So we have these different student ideas. We now need to construct actual questions or items that can tell us what the frequency of these different conceptions are in our larger student sample. There's a lot of different ways we could have done this. A lot of faculty in our department wanted us to do oral interviews, which would have been great. It has a lot of advantages. It certainly has a high resolution of knowledge, a high ability to pinpoint areas of difficulty. There's some issues with logistically, like how do you actually administer that? And also how do you, if you're going to do this in perpetuity or at other institutions, how do you have reliability or some sort of similarity to the way this would be structured? Generally, there's this sort of inverse relationship between the resolution of knowledge that you get from a particular format and how easy it is to administer. So what we settled on ultimately was this format called multiple true-false, which we think gives us a little bit more information than multiple choice, but it certainly is not reaching into these higher realms here. So the multiple true-false format is really very, very similar to a multiple choice question. In that a multiple choice question, you start with a question stem, which introduces some sort of scenario. You then have a series of statements. and students pick one statement that they think is most true. Multiple true-false structure is nearly identical. There's a question stem, and then there's a series of statements. The difference is that students are evaluating whether each statement is true or false. And this is important because we know that student conceptions are not always coherent, that they hold mixed conceptions of biological concepts, and that if they pick A, with the multiple choice question, if they pick A and A is true, we essentially don't know what they think about B, C, and D. We have no information on that. So the multiple true-false format has the advantage that we can actually force them to evaluate each different idea independently. And so I think it changes it really from a comparative task to at least some sort of an independent evaluation task. I'll call it semi-independent. These are still linked in that they're under the same question stem. But you could remove any one of them and the question would still function. There's also some research that the multiple true-false is more equivalent to free response item difficulties, depending on how you score it. And then I'll also say on a practical level, I think it's a lot easier to write multiple true-false questions. So multiple choice, you have to come up with one completely unambiguously true, correct statement, 4 plausible but incorrect statements, and that's fairly hard in biology. So this gives us a lot of flexibility to ask as many or as few true or false statements as we would like. Here is an example of a question that we developed. So the question essentially reads, there's a population of cells over here, there's another population of cells over here. These cells are secreting a signaling molecule. These cells have receptors that can bind to that signaling molecule. What are different ways that that molecule could get across this fluids-filled space? And then each of the different true-false options key in on one of those student ideas that were generated from the student interviews. So the first gets at this idea of the molecule having some sort of intuition about where it needs to go, or the receptor knowing that it needs to pull the ligand across the space. Questions about electrostatics and charge interactions, a motor protein being the vehicle by which this moves, and then finally, this is our attempt to get at random diffusion, the idea that the ligand can move across a distance, sometimes towards, sometimes away from the receptor. All right, so we have draft questions. We go through a very extensive revision process. We give them to students during Think Aloud interviews where they read each multiple choice question. They answer out loud for us, either true or false, and then they provide us some sort of reasoning, or they kind of talk through their reasoning on that question. This allows us to understand whether students interpreted the question correctly, whether there's any issues with language, and also, importantly, whether students are providing valid reasoning for their responses. So they might answer true and be correct, but for entirely incorrect reasons. And so this at least helps us to mitigate some of those issues. No question is ever going to be perfect, but we at least try to get a high fraction of students' answers we think being reflective of what their actual underlying reasoning is. We also give the questions to different faculty. I think each question has been through something like around 20 student interviews, around 20 faculty reviews. And with faculty, we're asking things like, does the question adequately address the stated learning goal? Is the question scientifically accurate? And we also ask them, is the question something that you think graduating molecular biology majors should be able to answer? The final step, once we come to sort of stable questions, is that we administer a pilot to a large group of students and measure the test characteristics. So the pilots that we gave were done in the spring and fall semesters of 2013. They were given online towards the end of the semester. Students received only participation credit. They did not receive credit for actually having correct or incorrect answers. And the final test has 18 questions, 4 statements for each question, and so 72 total statements on the assessment. And I will try to say, when I say the word question, I'm referring to a question stem and its associated true-false statements. When I say the word statement, I'm referring to an individual true-false statement. Our student sample, we were able to find 9 upper division classes at 7 different universities. We had an overall 86% participation rate. So 86% of the students who were sent surveys completed them. This is 583 students, and out of these 583 students, about 75% of them are seniors. The other 25% are juniors. There was like 1 or 2 sophomores, 1 freshman. So So in terms of getting students to actually take the test, I think we were successful. In finding a high proportion of upper division students, I think we were also successful. Now before I start talking about results, I need to clarify. So the multiple true-false format can be scored in a lot of different ways. There's people research different ways of scoring multiple true-false questions. The two that I will talk about, mostly I'm going to talk about what we call the fractional scoring method. And this is where students receive partial credit for each true-false statement that they answer. So if they answer 2 of these correctly and 2 of them incorrectly, they'd get 0.5 out of 1 possible point for that whole question. What I like about this is it retains all that information. You essentially get a score for each different statement. You're not consolidating or losing any information. The downside is that it has a really high guess rate, so it's a guess rate of 50%. The alternative way to score multiple true-false questions is what we call the all or nothing method. This is where students have to answer all 4 of these correctly in order to receive credit for that question. And if you're a student, obviously you would hate this because it'd be very hard to get all 4 correct. The benefit, though, is it has a very low guess rate. So it almost completely erases the guess rate. But like I said, you wouldn't want to do this to your students. It would be sort of a more challenging way of grading it. All right, so we gave this assessment to our large group of students. And this histogram is showing the distribution of overall scores on the assessment by the two different scoring methods that I just mentioned. So with the fractional scoring method, we see that students, there's a mean of around 66%, meaning they're getting 66% of those true-false statements correct, with a sort of a bell-shaped distribution, with certainly a fair amount of students down here around the guess rate, and students barely kind of popping into the 90s, but that's fairly uncommon. With the all or nothing, when you switch to this more strenuous way of grading, the distribution shifts down here. So the mean is now at 24%. You see very few students doing much better than 50%. The guess rate here is, I think it's 6% if I remember correctly. The math's not that hard, but I'm not going to do it right now. But this is, I guess the important reason to show this is that, generally speaking, students are not getting all 4 statements correct. Okay, so they're generally getting somewhere short of 4 statements correct 75% of the time. We can also look at question difficulties, and this can give us information about student achievement of these various learning objectives. So this plot, I have question numbers on the x-axis, difficulty on the y-axis. Keep in mind that difficulty is inverse from what you would intuitively think. So questions up here are actually the easy questions. These are ones that students do very well on. Questions down here are the ones that students that are much harder for students to have a lower percent correct on. Here I've separated out. This is the black dot is their score for the question as a whole. The gray dots are their percent correct for each individual statement. And the D is sort of hiding right behind the C there. You just can't see it. And this ends up being essentially a weighted composite of these individual. It's not even weighted, it's just the average of these ends up being this. So we see really a vast range of different difficulties. We see questions that are fairly easy. Up here we see questions that are particularly hard, well below the guess rate. One thing I'll point out here is that we're very concerned about whether the students are taking the test seriously and whether they're just blazing through it and guessing. Things like this where there's a high percent correct and a very low percent correct gives us a little bit of confidence that they're not just blazing through it. They're at least taking the time to read the question and if for some reason picking an answer that is not at random. We also care about reliability metrics. So we looked at internal reliability, which is essentially the covariances between different items on the assessment, and with the idea being that you would expect that there'd be some sort of consistency with the different items and that they would all sort of covary if they're measuring similar things. we achieved an alpha of 0.81, formerly known as Cronbach's. I've been told that you're not supposed to call it Cronbach's anymore because he actually didn't discover it. So alpha 0.81, which is on par with commercially available tests like the ETS proficiency profile, which is sort of a general test used at the college level, has alpha of 0.78. We also measured test-retest reliability, so this is the consistency of scores across time or across multiple consecutive administrations. The way we did this was we had one course where there were two different classes given in consecutive semesters taught by the same instructor with fairly similar curriculum. What we found was that the means were not different between these two semesters. And more importantly, when we look at the actual statement difficulties, so the actual percent correct on each statement, they're very consistent across semesters. So you get a correlation of about 0.91. So a statement that shows up as being correct about 60% of the time during semester 1 will typically show up as about 60% in the second semester. So we would say this capstone assessment has sort of acceptable reliability metrics. We can also drill down at the individual question level, and this is an area that we still need to do more work, but we can look at patterns of student answers within individual questions, and we see a couple of different patterns. So one is where there's one particular statement that's easier and students do better on, and then there's maybe 3 that are more challenging. We see the inverse where there's 3 that are maybe a little bit easier and 1 that is more challenging that students are not getting, so they're almost mastering this concept, but they have one area of weakness, and then pretty much everything in between. What I like about this is this then gives us information that we can use to design sort of follow-up exercises, and we have some idea of what aspect of that concept is actually causing students trouble. In general, I think we can say that this format at least allows us to conclude that students generally have mixed and partial conceptions of most concepts. And like I mentioned, this information can be used by programs to actually pinpoint areas of difficulty. Just to give an example, so the question on diffusion is one that Joe Dower and I, who is formerly a postdoc here at MSU, are very interested in. We've been designing activities for our introductory molecular and organismic biology courses to address this. And we hope that we identified a range of different incorrect conceptions from this test, and we hope to sort of pursue these on our own as well as other people in the field. Now, what I think is really interesting, this is where I think it's, I'll just show you the data, but there's 3 questions on the test that are essentially multiple choice questions sort of dressed up as a multiple true-false question. So they have 3 false answers and 1 true answer. And so they could essentially be a multiple choice question, and you would pick this and you would get the question correct. So what I wanted to know was, of the students who answered this correctly, what fraction of those students would get one of those other false statements incorrectly? So they would incorrectly answer one of these as true. And so what I found was that 88% of students, so of the 71% of students who answered this correctly, 88% of them will additionally pick one of these as being true, which I think is a strong sort of implication of the multiple choice format, in that we really cannot make any assumptions about what students know about these answers when they pick this answer. Another question you could ask is, on average, how many of those statements will they answer correctly or incorrectly? So for this question, it was about 1.3. So they're at least answering 1 or more. I'm beeping. I don't know.
I just plugged you in just in case.
Okay, thanks.
We don't have a meltdown.
So they're at least answering 1 of these statements incorrectly. We can do this for the other 3. What we find is that these percentages are still sort of alarmingly high, up at the 85, 70% range. And on average, they're answering, say, at least 1 of those statements incorrectly. So from all of this, sort of a tentative conclusion tentative interpretation would be that we would say advanced students are showing signs of struggling with fundamental biology concepts. They're not, the guess rate on the assessment is 50%. They're scoring at a 66%. That's not quite where we would maybe want it to be for our graduating seniors. The huge problem, though, is that the assessment was given under low-stakes conditions. And so is the data really a valid representation of student ability, or are they just kind of blowing it off? So this was a question that I wanted to investigate. And keep in mind, we're working under this major limitation that the assessment stands outside of any one single class. And so we really have not yet found a way to credit correct answers without getting into issues of test security. So if you make the test high stakes, you now have to secure the test. And we've not been able to figure out how to do that appropriately. So this limitation of having a low stakes motivational system, we haven't been able to change or study. I hope that somebody will consider doing that. What we can do is we can think about how and where we administer the assessment. And right now, the data I showed you was administered outside of class time. And we did that for a particular reason, because most faculty are not able or not willing to administer a test in their own class. They're short on time. The content is more than what's covered in their class. They don't find it ethical to use class time for this purpose. Understandable. There were a few faculty, however, who were willing to do this. So the question that I'm going to ask is, how does assessment location and formats, we're changing two things, affect student performance? The two options, one is to take it online at home, which is what I showed you before, and then the other option is to give it in a paper-based format in class. So the question is, what do you predict? How do you think the responses, the correctness will change when the same assessment is given in class? Let's see, how am I doing on time? All right, I'm just going to keep rolling, but if this were a class, I'd have you vote, and I'd have you talk to each other. But think about it. So we did this in 3 different courses. So these are courses where the same instructor is teaching back-to-back courses, either a semester or a year separated. They don't have any overt things that they would think are different about their course. Certainly there are differences in these 2 paired classes, but nothing overtly obvious. So these are our 3 pairs. First semester or second semester, this was inverted, depending on the class. It was given in either an online or paper format. First thing I want to look at was whether participation rates are different. So do you get a different number of students taking it when they give it in class versus when you give it at home? We had pretty high participation rate at home, and we ended up having pretty high participation rate in class. And these don't end up being very different. So up in the 90s, fairly consistent across the different formats. So that's encouraging. We also wanted to look at overall class averages. So how do students actually score on the test? So here are our 3 courses. In gray is the online administration format. In white is the paper-based administration format. And what we see and what the statistics support is that we don't see any gross difference in their overall scores. We do see a main effect of course, so there are differences between the courses, and that is definitely something that we saw earlier when we had a lot more courses. This test will distinguish between different courses, but we don't see any effect of format, and we don't see any sort of interaction between format and course. So this was surprising, because at least the people who I was talking to thought that the paper-based format in class would be much higher. That students, when they're on their own at home, wouldn't give it sufficient time, wouldn't give it sufficient energy, and that their scores would be really low as a consequence. So when you give students the space in class, their performance is not overall different than if you give it to them at home. We can also look down at the individual statement level and look at the consistency of scores on individual statements. And what we find is that they are very closely tied. If you were to do a correlation of these in the same way that we did it for the test-retest reliability, you'd see that there's an identical or essentially the same correlation between statements. So the differences that you're seeing from format are no greater than the differences that you would see from simply one course being given in subsequent semesters under the same format. I did do a Mantel Hinchel item bias test, which is sort of the gold standard for looking at item bias between different formats. There are items on here where I would have expected there to be a difference. So there's one where students have to translate a sequence, and I would have expected that they would benefit from being able to write that sequence down on a piece of paper. That item does not show up as being different between the two formats. There's another one where they have to draw a Punnett square. I cannot for the life of me do genetics without drawing a Punnett square. Students seem to be able to do that, or they seem to not be willing to draw a Punnett square. I have looked through the test booklets, and I haven't quantified this. We don't have the test booklets anymore. Students generally are not writing on these tests. So that might explain why some of those items that we would have predicted to be different are not different. There are a few that do show up as being statistically different between the two. And this test takes into account sort of overall ability, and then it's sort of like a chi-square where there's an expected value and an observed value, and it looks for statistically significant differences. I've looked at these. There's not obvious reasons why one format would be higher than the other. There's at least one where it's really sort of a trivial question. It's something you could look up on the internet very quickly. That one's a little higher in the online format, so maybe it's something that's so easy to look up, students would just look it up when they have access to a computer. But otherwise, it's not obvious why one item would be higher than the other. So from this, we know that essentially 68 out of the 72 statements are fairly consistent between the formats. Some of the statements do show some bias. And the other thing that's interesting is if you look here at the end, you see a little bit of tailing off. So that you start to see some separation here, with the paper version being a little bit lower than the online version. So another thing that we looked at was just raw question completion. So this plot is measuring for each question how many students actually put an answer. Right or wrong, just actually answered the question. And with the online format, we see that we have roughly almost 100% completion across the entire assessment. With paper, we see that towards the end, there's a tailing off, starting around question 13 or so, and reaching up to about 10% of students not completing the last couple questions. So this can explain the gap that we saw in the previous graph between the two formats. But two, it also points to the sort of inherent limitations of giving an assessment in paper in class, particularly when a faculty member does not highly value that assessment or see it as part of their normal course. So in one class we had, this is a composite of 3 different classes. In one course, the tailing off is relatively minor. In one course it's about like this, and in another course it's much more extreme. So you actually also see variability. This is a big issue for test developers. So you don't want an administration format that's going to have a variability that's linked to particular items. You might see a low score on an item simply because you did not give enough time that particular year. And while the faculty were very helpful and willing to administer this for us, I mean, we couldn't have done this without them, they were just under these legitimate constraints that I have some material I need to cover. I'm going to try to get this assessment in. I'm going to give it 30, 40 minutes, and when students are done, we've just got to collect it. Class is over. So what I would say, we spent a lot of time debating about whether we should give the assessment online at home or on paper in class. And the paper format is really sensitive to the amount of time that the instructor allows. And I think you could make a case that the online format, for that reason, is actually preferable. Another question, we can track how much time students spend on every question, on every page, and we can sum those together. And this data is a little bit filtered, right? Because students, we don't know what they're actually doing when they take a question. They could start a question, go eat a sandwich, and come back, and it'll look like they spent 40 minutes on the question, and they probably didn't spend 40 minutes. So I do process the time data by capping every question at 20 minutes. So once they spend more than 20 minutes, their value gets reduced down to the mean for that class. So these have to be taken with a grain of salt. But this is the distribution of how much time students appear to be spending on the assessment. Each number refers to the amount of students who are spending up to that amount of time. So students are taking, there's about 10% of students who are spending less than 15 minutes. We would say that that's not enough time to complete this assessment. The in-class version, we've never seen anybody finish in less than about 18 minutes. So we figure that assessment really takes somewhere between 20 and 40 minutes, which is where we see the bulk of our students actually appearing. Then there's students out here who are taking longer. We're not sure exactly what they're doing. But at least, say, starting here, about 80% of students are spending 20 minutes or more on our assessment, which is what we would hope. So sort of the grand conclusion from this is that under the given low stakes conditions, which we feel is a constraint that we haven't been able to solve, the online outside of class is comparable and perhaps better than, because it eliminates the time issue, than an inside of class format. And also, I don't think it's so easy to dismiss our early results as simply being students not caring at all. I think we have to realize that these are comparable to what they would give us if they had the time and the space to give the assessment. So this administration format issue is important because we are working to develop more assessments. We have been given an award from the NSF to develop what we're calling biomaps assessments. The issue with the Capstone assessment is that it was built for graduating students. By the time they get there and they graduate, if we find deficiencies, they're gone. We can't correct those deficiencies. So with biomaps, we're hoping and working on developing assessments that would be able to be administered at an entry point, some midpoint in the curriculum, and then as well as when students leave the curriculum. And then we can actually use that formatively to influence what's being taught here and here. So the assessments would be administered at multiple points during the major. Whereas the Capstone assessment was restricted to molecular biology, we're trying to cater to all the different types of biology departments that exist, including general biology, molecular biology departments, physiology, and also ecology, evolutionary biology. This is a big collaboration between lots of folks at lots of different institutions, and we're actually piloting like right now as we speak. So I'm excited to see that data come in. All right, so I'll be the first to critique this data, but here's some of the things that I think we can learn from it. And this is sort of my best shot at sort of a nuanced message. So one is that I think there's something to be said for thinking about assessment at a programmatic level. So while the stakes, we've had issues with making these assessments high stakes, I wouldn't say that they are inferior to in-course assessments, in that in-course assessments also suffer from limitations, in that you have to, there's a sort of social contract where you have to tell to some extent students what's on the assessment, or at least that's the assumption or the implicit contract. They're able to study very specifically the night before. We have no idea what happens after they take the assessment, whether they retain it or not. I'm certainly not advocating that we replace course assessments, but I think this is an alternative view that we ought to be thinking about as educational researchers. Also, at the departmental level, thinking about our program as a unit rather than as a series of discrete courses, I think is valuable because it sort of paves the way for us to think about cohesion and sequencing of our courses and the topics that are taught therein. The sort of tentative conclusion, or the most simplistic conclusion, would be that advanced students are still struggling with fundamental biological concepts. Now, I would qualify that by saying, one, that the test was built to find things that students are struggling with. There are not a lot of, softball questions on there. We're looking for where students struggle. So that was an objective of the assessment. But what I do think there might be is a misalignment between what faculty expectations are and what actual student learning is. Generally speaking, our faculty, above 90% of faculty said that these questions were things that biology students should know when they graduate. And there's questions where they're sort of horrified that their students would answer at such a low rate. And I think that's sort of the discussion that we need to be talking about is what are realistic expectations for students, and are they achieving those expectations or not? I think generally students seem to be exhibiting mixed and partial conceptions of different biology concepts, at least within an individual question. This is consistent with other research. But the point I would make is that I think multiple choice in particular is ill-suited to pick up on these mixed and partial conceptions, and might actually be overestimating in more than one way the students' conceptual mastery, and that the multiple true-false provides a convenient and not so different way of asking a similar question, but getting a little bit more information and accounting for this element of student thinking, that it's mixed and not completely formed. And finally, I think there's a tendency when people talk about programmatic assessment, they see it in sort of all or nothing terms, where we can either do it whole hog, where we require, we make it worth a course credit, and if some students don't do well, they have to do some remediation course, or we don't do it at all. And I think this research at least points to the idea that it's feasible to do an outside of class administration. It's comparable to what you would see in class. It's certainly not high stakes, but my hope is that programmatic assessment will be used in more of a formative way, and I still think that the results that we're seeing can be used in a formative way. We can still identify which concepts are particularly difficult and which ones are not as difficult, and that can help guide our efforts. So I think we should think of this as an open area of research and not one that we should disregard because there's some serious structural issues in play. All right, so with that, I'd like to acknowledge all the people who helped with this. So now at Nebraska, I have my own shop. I have 2 postdocs who are working with me, and I'm also hiring another postdoc if there's anybody who's interested. And then we have the pleasure of working with the Dowers, who, as many of you know them, they're great colleagues. Jenny Knight was my postdoc advisor, and she contributed extensively to this work, and she was a pleasure to work with. And then our collaborators at other institutions and our funding. So I'm happy to take any questions. That's all I have for today.
Have you attempted to look at any of the courses or even half of the courses in the sequence for the students that follow through that 4-year trajectory, if any of them really have much impact on performance on your test?
So you're saying students who...
Like, for instance, maybe students who took none of the upper levels, do they do differently than those that took upper levels?
Well, so partly this is an agglomeration of multiple institutions. So tracing, at other institutions, we don't have the ability to get that registrar type data. So we don't see a difference between juniors and seniors. So juniors and seniors are not different. And it's not clear whether we would expect that. Because juniors who are in a Capstone course and seniors who are in a Capstone course may still have had the same, coursework prior. What we haven't, some people are very interested in pinpointing like which courses, is there a particular course that's associated with low or high performance? That's not a road that we've gone down too far. It's very contentious. So I don't know if I could. That's really all I can say to that, I guess. Yes.
So could you talk about the content of the test? Like, we got a bunch of people here to teach cell molecular biology course.
Yeah.
Suppose you're going to them and you'd say, this is what students are having trouble understanding. What's the this?
Oh, I know. I wish I've been searching for that singular thing. We've done factor analysis and we've identified individual sort of somewhat discrete factors that we think are embedded in this test, one of which is evolution, one of which is along the lines of the information flow, exchange, and storage. I can say that in particular courses, so when we do sort of factor scores at an individual course level, we will see differences. So one course will have a very high evolution score and will have a very low evolution score. Presumably that's due to something that's in their curriculum. I don't know if I could distill it down to like this is the problem. We have the assessment. It's published. You can see it, and we also wrote. This was actually at the urging of Mike Klimkowski. What do we call him? I guess we call them knowledge statements, but all of the information that you would need to answer every single question and statement on our test. And embedded in there would be the idea, these would be the things that students do not know as evidenced by the performance. But
So you have knowledge statements that are particularly difficult for the students?
Yeah, so I can show you, okay, so I can, so in a biology audience, I typically show this slide. So these are some incorrect ideas that biology majors endorse. So these are called basically if more than half of students endorse them. One, the idea that inbreeding causes new alleles to develop in a population. I view this as a somewhat of a discrete thing, and I have not been able to connect that to a larger, mental structure. One, in developmental biology, the idea that cytoplasmic factors and unequal distribution of cytoplasmic factors is an important way of differentiating cells. This is sort of classic from the biochemistry field, the idea that enzymes promote catalysis by raising the free energy of reactant molecules rather than affecting the activation energy. Again, this is down in the weeds, so binding affinity between two molecules, and this is for non-cooperative binding interactions for people who care about biochemistry, changes as a function of their concentrations. The idea that a protein that's binding to some target that's present at a higher concentration and is binding, it has a higher binding occupancy, not binding occupancy, but its fractional saturation is higher, is because it has a higher affinity. It has the same affinity, it's just there's more of it around, and so more of it combined. And then another one, this is one that I think gets at a broader issue, the idea that SNPs, these are small changes within the genome, that if you have a SNP that's associated with a disease, that SNP is directly causative of that disease. And that's sort of a specific question, but it gets at this bigger issue of genetic linkage, and that genomic regions can contain markers that can tell us information about that entire region and not the actual problem. So these are some of the statements that just came out as being particularly difficult. But then there's a whole range of things that are in the, just over half of people get them correct. They're like in the 60% range. I would still say that's not adequate. Yes.
Well, first, I just want to address that if when you say over half of them get them correct, but half of them could be guessing.
Yes.
10% getting it correct. Right?
Yes.
So since they've only got two choices.
Yeah.
And I want to go back to this idea that you're assessing your learning objective, where it's the one you showed, where it's to explain how molecules move around. But they're not.
They're not.
Right? So
I agree.
it's and even students, as you pointed out, even students who get the right answer probably can't do that anymore.
Yeah.
So have you thought about in your new project moving forward from thinking only about what students know, but to what students can know and can do with the knowledge?
Yeah.
Right now you're still addressing snippets of knowledge, right?
Yeah. Okay, so what are those different things I think about? One thing I totally agree and concede that they're not explaining anything, right? And it is not a substitute for explaining, and it's very cognitively different. Just to highlight one benefit, though, between multiple true false and maybe more of an open-ended thing. So in interviews and with open response questions, students are unlikely to address all of the points that we might be able to ask in a multiple true false question. So they might give a correct answer and completely not mention any other area. And so this at least allows us to probe that area in a way that's hard to do, because with an open-ended prompt, students might just not think to answer that. But is that sufficient? No, I don't think so. With biomaps, we're still using the multiple true false format. We are the type of question in terms of focusing on concepts rather than on scientific process is still going to be the same, partly because we're aware that the University of Washington is developing a science process skills assessment, which that's not to say that that's the end of science process skills assessments. You're talking more about what they can do in terms of...
Well, I think if the two are separated, then that's very different.
Yeah.
Doing something with their knowledge. So I'm not talking about science process
It's-
skills, I'm talking about
Okay.
the integrated.
Yeah.
Can they construct a model of how molecules move across the system?
No, I don't think we're not capturing that.
Okay.
I don't think, I mean, I would love, I don't know if, maybe a way of getting at this, I think doing that at the programmatic level is, just getting this into a program, it was challenging enough. I am interested in doing that sort of stuff on a course level. I think that's a place where you could feasibly administer those kinds of things and get information. And I'm in no way discouraging that at the programmatic level. I think this was sort of the first pass, the thing that we felt like was feasible. So, yeah.
One of your first goals was to encourage conversation at the department level. Can you tell us anything about how what is this encouraged?
Yeah. I think that's the most challenging part, right? Is to take a critical eye towards our curriculum and to take not just a critical eye, but an inquiry into our curriculum and what it's doing. In terms of the conversations, I think there was some of our discussions did generate crosstalk across courses. So, I teach this course. I thought you had taught this before, and you're telling me now that you did not teach this before. And so there was a little bit of sort of where do these things go in our curriculum, and are they being taught at all? But nothing systematic, right? There was no grand, chart looking at that. I'd say on the other end, now that we have data, it's, I think it's going to take time, honestly. Most of the conversations I was involved with were still wrestling with, is this data worth anything, and should we be spending the energy to change things if the scores are artificially low and actually our students are quite proficient. So since having left now, my understanding is that they are actually taking this up, and they're going to continue to give this assessment. I know that in the undergraduate education committee, which I was a part of, we did talk about these results. We did try to think about what can we do in our curriculum to change them. Going back to this first question, though, we didn't have a grand solution. So there was no easy, if we just get this one concept really down pat, this would allow all these other concepts to be understood well. So it's not, there was no quick or easy solution that we could identify. Do you have a question, Caleb?
Yeah. So, as a graduate of that program, I kind of had a similar question. And just so how are instructors using this information? Is there any talk about, making some accountability, either by the students or by the instructors? Similar question, just who's benefiting from this data?
Yeah. I think on a student level, I don't foresee accountability you need to do well on this and demonstrate that you've learned these things in order to get anything. People have talked about paying students for correct answers. They've talked about getting graduation cords for correct answers. They've talked about all kinds of things. And fundamentally, this test is on the internet. As soon as we bump up those stakes, the test is going to be compromised. And I think we kind of lose sight of the purpose. On the faculty side, it's also problematic in that departments are going to be reluctant to give an assessment that implicates individual professors, individual courses. And I don't think that's in keeping with our spirit of supporting our faculty, right? So I think it's really on the faculty themselves to derive inspiration from this and to have conversations about it. But I don't see there ever being a strong accountability.
But are there instructors who are?
Yeah, I would say Yeah, I'd say the people who partnered with us in the development are interested in the results, and the people who didn't partner with us in development are not as, But we had a grand faculty meeting at the end, right before I left, and they were interested. There's so many constraints in terms of time, and teaching assignments change all the time, but their ear is on the rail, and I think they're cognizant that there's some things that they could improve. But in terms of concrete stuff, I can't point to like a concrete thing. I'm certainly trying to do things, but it's hard on others. Yeah.
I'm always interested from a methodological point of view when folks choose to use classical test theory sort of approaches as opposed to addressing some of these things through IRT or Roush. Did you have a big enough sample there? Was that a decision in part because the classical stuff is a little bit easier to explain to the folks you're working with, or are you looking at these things with some IRT models?
I have done Roush modeling. The sample size, it depends on who you talk to, whether it's sufficient or not. So my understanding is that 300 is sufficient. I've also just recently talked to a psychometrician who's saying that you need 1,500 data points. And it's like, well, which one is it? So we're somewhere in between there. The things that I see in terms of whether the test is, whether the individual items fit within the model, my sort of conclusion was that the items that were outside the boundaries of the model were very much the items that had low discrimination or very high discrimination. And so I didn't see the overall conclusions of which items are better items and which items were maybe not psychometrically as sound didn't change a whole lot between classical and IoT-based models. We do have some low discriminators, some ones that are outside that don't conform well to the IoT model. We've done a lot of student interviews with those and with very close attention to whether students are interpreting them correctly, and we don't find issues. And since we think there are things that are problems that we should know about, we've left them on. But that's a limitation of, I view that as a limitation of the test. Yeah.
In the stage when you were doing interviews, did you find, and you were looking at those sort of vision and change categories, did you find any insights into problems that students were having in any one category?
Let me think back a little bit. I think so these were done at CU. Generally speaking, evolution is not, and these are molecular biology students, and they don't ever take an evolutionary biology class. That was something that was particularly problematic. So there's a question that doesn't use the word genetic drift, but it gets at genetic drift, and it was like, nothing. Students never even conceived of this. That was certainly really anything around evolution was a problem, except for the very most superficial sort of natural selection question students at CU struggled with. I would say also protein DNA binding was challenging, and also not at a...
In the structure function side or in the information side?
In more of like a... almost more of like a... Not really a structured function, more of a... like understanding the dynamics of protein interactions with their target, or any binding interaction, and how that changes as a function of concentration, how that... the idea that interactions are transients, that sort of thing.
So sort of the basic chemistry.
Yeah.
Sounds almost like it's getting at systems level thinking there.
Yeah. Systems we really have wrestled with, because it's hard to get at this idea of a system in like a multiple true-false statement. I guess the idea that you could combine a lot of different multiple true-false statements, and that might start to probe a little bit at systems. But as soon as you reduce it down to a statement, it becomes more about information flow and less about a system. So that was an area we struggled to actually get into.
So given that we're almost at 1:15, I think we should thank Brian. We have a little parting gift. Everybody gets a mug, but not everybody gets a bag. So I think you should feel a little special about that.
Wow, thank you. And I heard that I'm responsible for lunch, so you're welcome for that.
Yeah.
That's very nice. Cool.
Thanks all.